So let's go. The acid-base character of amino acids. No, the structure of amino acids. We know amino acids contain an amino group or NH2, right? And our carboxylic acid group or COOH. Amino acids will undergo most of the reactions of amides and carboxylic acids. Guess what? Because they are amides and they are carboxylic acids. Interesting, isn't it? So apart from guanine, all right? No, it's guanine or alanine. Apart from guanine, I think. All right. Apart from guanine, there's something that is interestingly observed in these molecules. Can anybody tell me? There's something interesting about amino acids. Nobody can tell me. Look at the structure, man, and tell me what's interesting about it. Not one person want to give it a go. Tell me what's interesting about this molecule. Is it that chemistry is just that disinteresting? It's not that bad. All right, I guess so. In the interest of time, notice that here is a chiral carbon. These compounds can form enantiomers and diastereomers. Amino acids are optically active, right? So, unless you're guanine, then you're kind of boring because guanine's R group is just a hydrogen. Therefore, we won't have chiral carbons. But yeah, it's actually optically active. All right. So you can have like resymmetry and stuff like that happening, right? So all of them except for guanine with the boring self. No, it's not guanine, it's glycine. See here, glycine. Glycine doesn't have a chiral carbon because it has, two hydrogens. So it's a carbon attached to three different groups, so it's not chiral. But apart from glycine with its boring self, everything else, right, is displaying some amount of chirality, right? So let's have a look at this now. So we have acidic, right? So these are the acidic groups, right? So the acidic groups are over here: the alanine, valine, leucine, methionine, isoleucine, phenylalanine, tryptophan. These other amino acids are all acidic. Then our histidine, arginine, and lysine are actually our basic amino acids. Then we have neutral nonpolar, so tryptophan, glycine, alanine, valine, right? Proline, these are all neutral nonpolar. But our polar substances are tyrosine, our asparagine, our glutamine, our cysteine, right? Well, your cysteine actually, right? These are polar, right? Just looking at the different amino acids, right? So the Zwitter ion. Amino acids are Zwitter ions due to their unique chemical structure, which contains both acidic and basic functional groups. The amino groups consist of a central carbon atom, the alpha carbon bonded to four different groups, a hydrogen, an amino group, well, carboxyl group, and a variable side chain, the R group. In a neutral aqueous solution, the carboxyl group tends to lose a proton to become negatively charged. We looked at all of that, right, to form the carboxylate ion. Meanwhile, the amino group will become, right, NH3, will form an ammonium ion, right? So it will be like that. So both groups will cancel each other out because one will become negative, one will become positive in a neutral solution, right? But the net results actually change depending on the type of solution it's in, acidic or basic solution, right? And we need to talk about all that internal ionization, right? So once you have internal ionization, right, which could be negative or positive, that's why we call it a Zwitter ion. It can bear both a positive and negative ion at the same time. Okay? So the ionic properties of amino acids. Amino acids contain the acidic and basic group as we talk about, right? Amino acids are usually ionized at physiological pH, which is around 7.4. Our body is alkaline. It's not neutral, right? Our body is alkaline. Therefore, amino acids are amphoteric, right? So they have amphoteric properties. So in acidic medium, the amino acid is positively charged and behaves like a base, right? So a proton acceptor. And in alkaline medium, our amino acid is negatively charged and behaves like an acid. So it is a buffer. And this is what we talked about in unit 1. We talk about buffer systems and the fact that protein buffers use this type of system, right? And here we can see the example of the amino acid zwitter ion. Okay? So in neutral solution, for example, water, both groups are ionized, and the positive charge on the ammonium ion, right, balances out the negative charge on the carboxylate ion, and the solution is electrically neutral. Everybody get that? Everybody understand that? Everybody get that, right? Yes, no, maybe, kinda, probably. All right, I'm gonna assume that everything is fine. Okay. In acidic solution, the acid, right, provides hydrogen ions, right? So for example, in hydrochloric acid, right? So the lone pair on the nitrogen atom, right, the NH3 plus group, is already protonated in the Zwitter ion. So the addition of a hydrogen ion keeps this group positively charged, and the COO group ion is a proton acceptor, right? So therefore, that carbonyl group would actually accept a proton, and that means that the amino acid, right, would actually be positively charged, because the amino group still is positively charged, right, while the negative charge of the carboxyl ion is diminished by the addition of a hydrogen. So overall, the solution is electrically positive. Make sense? If it not make sense, let me know. If it not making any sense, let me know. Okay. In alkaline solution now, the alkali provides hydroxide ions, right? For example, from sodium hydroxide, which is the most basic alkali out there, right? So the NH3 plus group is this in the Zwitter ion acts as a proton donor, so it gives off its proton to the hydroxide, right? While the COO group remains negatively charged, right? Because it's not gonna try the solution itself is gonna be actually electrically negative, so it can't really accept extra protons because the hydroxide ion sucks up all the extra protons. So overall, no, we're gonna have electrically negative solutions. And the compound itself is actually electrically negative. Alright? Any questions, any concerns? Alright? That is basically it, that is literally it. Talk about the formation of the sister ion, how it is in neutral solution, how is it in acidic solution, how it is it, or how it is in alkaline solution, right? And then we look at this. Look, a question, right? Don't get too excited now, right? The amino acids can be recrystallized from aqueous solutions. These crystals have high melting points. Give the fully displayed formula of the two isomers of alanine and explain why alanine has a high melting point and is soluble in water. Interesting. Interesting. Not sure if you're gonna be able to do that, but only the bravest of the brave may attempt this question. All right, that is it.